Differential reproductive success or natural selection changes our allele frequencies, and it is often the most influential change on allele frequencies in a population. So let's talk about a few different types of selection, or a few different ways that natural selection works on a population to give individuals differential reproductive success. So the first we're going to talk about is balancing selection. So in balancing selection, the heterozygote has a selective advantage. So both of the alleles are going to be present in these individuals, and those individuals are going to reproduce at a higher rate because of that combination of alleles. So this is a similar diagram to what we saw a few minutes ago in the last video, which was directional selection, where one type of individual is selected over the other. One genotype is selected over the other, and it shifts the population towards one genotype or the other. So in balancing selection, our heterozygotes are selected for. So in this example, we have individuals who are homozygous, only produce one offspring. Individuals who are homozygous for the dominant allele produce two, so they have a slight advantage over the other homozygote. But individuals who are heterozygotes produce twice as many offspring as the homozygous, and 4 times as many as the homozygous recessive. So in this case, we are going to maintain heterozygosity in the population. You might also notice that neither of these alleles can become fixed. The A allele can't become fixed. The little a allele can't become fixed, because both alleles are going to be maintained because heterozygotes have the largest advantage. An example of this is sickle cell. We have talked about this several times already this semester. Sickle cell in itself is a deleterious mutation, or a deleterious allele. So if you are homozygous recessive for sickle cell, you are less likely to survive to reproductive age and to pass on that allele. But if you are at such a disadvantage for having it, why is it even maintained in the population? Well, the answer is heterozygote advantage in regions where there is prevalent malaria. So it turns out that the sickle cell shape of the hemoglobin actually gives some resistance to regions where malaria occurs at a high rate. So in these regions, individuals who are homozygous normal for their hemoglobin are going to be or potentially be infected by malaria and potentially die from malaria. Individuals who are homozygous recessive are going to have sickle cell, and they are likely to die from that sickle cell. But it turns out that individuals who are heterozygous for sickle cell are less likely to die of sickle cell, because not all of their hemoglobin is sickled, and they're less likely to die of malaria. So it turns out that individuals who are heterozygous are just as likely to contract malaria, but they're less likely to die from severe cases of malaria, because the malaria Plasmodium falciparum, which is the protozoan that causes malaria, is less able to reproduce in those sickle-celled shape hemoglobin. So they're not any less likely to be infected, but they are less likely to die from the disease. So in regions where malaria is prevalent, the sickle cell allele tends to occur in higher frequencies because it does give a selective advantage. Now, of course, if you are full sickle cell trait, you are going to die of sickle cell regardless of your malaria. But that is why, even though sickle cell allele is a deleterious allele, it still remains in high frequency in parts of Africa and in other parts of the world, like the Middle East and India, where malaria is quite common. So down here on the bottom, it's just a diagram that shows the change. We've gone over this before a couple of times. It is a single base pair change that results in an amino acid substitution that changes glutamic acid to valine, which results in that sickle-celled shape. So individuals who are heterozygotes produce both the normal and the sickle-celled shape. Another example of heterozygous advantage is the CCR5 gene. So the CCR5 gene is a gene that produces a receptor on the outside of specific immune cells. And it turns out that individuals who lack the CCR5 protein receptor on their cells are resistant to HIV infection, because without that CCR5 receptor, the HIV pathogens, the HIV virus, cannot recognize the cells that it is going to infect. So it turns out that deletion in the CCR5 receptor has very little deleterious effect, and that deletion is actually quite common in Europe. Now, it seems that there is a heterozygote advantage here, but researchers aren't really sure what the advantage is. It seems that HIV is too recent to have been a selective agent throughout the larger evolutionary history of people. So it's possible that there's a heterozygote advantage due to some other pathogen. It was selected previously by another pathogen, maybe similar to the malaria example. But I find this interesting. This is something that I didn't know was a thing until recently. So I do find it interesting. All right, we can also have frequency-dependent selection. In frequency-dependent selection, the most common allele or the favorable genotype depends on which allele is the most common. So this is most common seen in host pathogen interactions. So basically, an allele becomes more frequent, and then it begins to be selected against because it's more frequent. And so then it goes down in frequency, which means the other one rises in frequency. And so once that one gets high enough, it becomes the selected allele. So our example here is we have these individuals who are resistant to a particular pathogen, probably because these receptors on the outside of the homozygous individuals make them susceptible. So, of course, those individuals that are resistant are going to increase in frequency in the next generation. Those that are susceptible are going to decrease in frequency. Now, this is all fine and good, except that infective agents are basically at an arms race with their hosts. And so if a pathogen mutates, a virus or a bacteria mutates to now be able to infect and identify and infect these cells that were previously advantageous, they are going to be selected against, and those without this new receptor that the pathogen does not recognize are going to increase in frequency. So this is sort of the back and forth, where if you have the right receptor, you're going to be selected against, but those are selected heavily against while the other ones increase, and now they become the most frequent. Then they are most heavily selected against while the others are not, and so on and so forth. So let's look at one more example of selection. This is positive selection, or a form of directional selection. So remember that directional selection selects for one genotype over the other, usually the homozygous genotype. Now, directional can also be negative. It can select, the opposite is that it's selecting against something else. But let's look at this example of positive selection, and this is lactose persistence. Now, lactose persistence used to be called lactose intolerance, suggesting that people who cannot process milk are actually the ones that have changed, that have become different. But in fact, throughout evolutionary human history, most populations were actually unable to process lactose past the point of infancy. So lactose persistence is where populations or individuals continue to be able to utilize lactose as a sugar source after weaning. So all mammals are fed by milk when they are infants, and then eventually lose the ability to digest lactose. But some populations actually have lactose persistence, where the ability to break down lactose in milk persists throughout their lifetime. So lactose is a common sugar in milk. It is broken down into glucose and galactose, which are more easily used in the cell. Now, this lactose is broken down by lactase, which is the enzyme that breaks the bond between glucose and galactose. It is produced from the LCT gene. So the LCT gene is often shut off at 2 years. So transcription factors block LCT and shut it down after about 2 years or so. And at this point, most other, in humans, it's shut off after 2 years. And in other mammals, it is shut off after the individuals are weaned. So at birth, the LCT gene is transcribed and is able to make lactase. Once it is turned off, the individual becomes lactose intolerant. If it remains on due to lactase persistence, those people continue to break down lactose throughout their lives. But people from some populations and not others continue to transcribe the LCT gene after weaning. And it turns out that those individuals come from historically pastoralist populations. These are populations who depend on pasture animals, cows and goats, things like that, animals that are producing milk. So this lactase persistence is traditionally common among pastoralist and herding populations, those that are raising cattle and goats and camels, things like that. So if you look at the distribution of lactase persistence, you see that it tends to be more frequent in locations where populations were traditionally raising cattle, raising goats, things like that. And they believe that this is because lactase persistence had a selective advantage in those regions. Because if you are around animals all the time, you have a dependable source for fluids, vitamins, and calories throughout your lifetime. So what likely happened is that some individual acquired a mutation in or near the LCT gene, and then because it was advantageous in these regions, it became more frequent within the population. But it turns out that the haplotypes, or the SNPs, that are related to lactase persistence are actually different depending on the origin of the population. So the lactase persistence mutations are found upstream of the LCT gene in the regulatory region. So this will be the cis regulatory region. And this makes sense because the LCT gene is already functioning and present in individuals, but the lactose persistence or intolerance comes from a change in the way that gene is expressed. So if that gene is going to continue to be turned on, transcription factors have to continue to allow it to be turned on. So this diagram here just shows some of the different populations that have lactase persistence, and they are all upstream in the regulatory region for LCT. Now, this suggests that lactase persistence occurred in these different populations separately. There are different sources of lactase persistence. So different populations have different single nucleotide polymorphisms and surrounding haplotypes than the others, but they all confer persistence for lactase. So each one of these mutations would have occurred as a selective and then have been selectively advantageous, in which case they became more common in the population. So if we go back to our map here, the highest frequency of lactase persistence occurs, the highest frequency of the allele occurs in these traditionally pastoralist regions. And then we have other regions where basically no one has lactase persistence into adulthood: Asia, Australia, Southern Africa.